Greetings, ladies and gentlemen. Today we are going to make a prokaryotic cell out of recycled materials, starting with styrofoam. I found this styrofoam at the box where our refrigerator was bought. Next is clay. We are going to use these clays to form some parts of our cell. We are going to need some glue to glue the parts of our cell. And finally, a cutter. Other materials will be used later in the video. First things first, I cut the styrofoam into two 16 by 9 cm measurement of styro. This will serve as the base of our cell. Next, we are going to glue them together. After 20 minutes, they are now bonded tightly. Now, we are going to trace the outline of our cell. Next is to cut the trace area of our cell. At this point, we are going to cut the edges of the styro to shape the base of the cell. It requires patience and accuracy to avoid breaking the styro and to evenly shape its base. Of course, I am having a hard time doing this because I do not belong in the arts program. But I am still trying my best because STEM people are versatile. Next is to cut two 9 by 5 cm of styro, then trace it to the edge of the cell. And glue them together. It is now day 2 of our project. We are just going to glue this part to the base of our cell. So I added another styro to elevate the cell. We will again cut the edges of the upper part of the styro. Next is to shave the upper part of the styro to form the cell. This process took a while though. I initially traced the cell, and now we're going to cut these areas to reveal its coating, capsule, cell wall, and the cell membrane. I also did the same thing to the remaining coating of the cell. Day 3 of the project. We want our cell to have vibrant colors, so we need to paper mache it. We need used papers to cover our cell and apply a solution of water and glue on it. This is how it should look like. I already made the flagella of the cell. Now is to make the pili or fimbriae of the cell using clay and toothpick. The cell uses this part in order for them to move. We will color the tail with green. After which is to color the capsule of the cell using red color. Yes, at this point onward is fun. Next is to color the cell wall with yellow. And lastly is to color the membrane into green. Color the cytoplasm with blue. Next is to make the DNA strand of the cell. And finally is to make some organelles like plasmid and ribosomes. Prokaryotes are unicellular organisms. They lack organelles or other internal membrane-bound structures. Therefore, they do not have a nucleus, but instead generally have a single chromosome, a piece of circular double-stranded DNA, located in an area of the cell called the nucleotide. Thank you for watching and peace!